Okay, good evening, all. Hope I'm clearly audible to you. So, welcome to the NPTEL course Immunology. So, I am Ananya Chatterjee. I'm a PhD student in ISC Bangalore, and I'll be your teaching assistant, one of the teaching assistants, because there are two teaching assistants for this course. So I'll be basically discussing all your doubts regarding courses along with every like assignments, and then exams, registrations and all. So you will be I'll be basically every week I'll be discussing the course content as well as old assignments like previous year's assignments and as well as this year's assignments. But one thing we need to remember that we have typically for every assignments we do have 14 days time to submit. So before that I won't be able to disclose answers to you, but I'll be happy to discuss if you have any doubt. But once the assignment date or the due date is over, then only I'll be discussing those assignments. Okay. And yeah, course contents is already started. So now week 2 is going on. Week 1 lectures and week 2 lectures has already been uploaded. So please go through all those lectures carefully, listen to those lectures, refer. I will also refer some books and then go over slides, and those slides also I will also go over in my classes, and others extra some like whatever questions you will be asked in assignments, those parts also I will cover. And so yeah, we will start today's class. But before that, as this is the first introductory class for me and for you as well, for the tutorial sessions, I would like to know that from which background you all are, and kind of introduction meeting we will have, and then I will start with week 1 content. So if you can use the chat section to avoid any noise. You can just type in the chat section that in which courses you are, and if you are in BSc or MSc, you can write down your like in from where you are doing, which university you are doing. So please feel free to write, then I will have a sense that in which course you are, BSc or MSc or BTech, MTech. Please start volunteering. Am I audible to you guys? Oh, nice. MSc Biotech, BSc Microbio, MSc First in Zoology, Wildlife Studies, Medical Electronics Engineering. Hi Akshata. I've heard this course name for the first time. Where you were doing IIT Kharagpur? Hello. Okay. Medical electronics engineering. I have heard this. The course name, it's interesting. I've heard this for the first time. Nice. So all over, you are all more or less from biology background, and then BSc and MSc or BTech, MTech. Okay. So I have. I will also give my introduction a bit. Now I am at third year PhD, and I'm doing in IIC Bangalore, and I have done my bachelor's in zoology from Banaras Hindu University, and I have done my master's in chemical and molecular biology from IIT Kharagpur, and I joined IIC for PhD in 2021. So you can discuss with me any doubt if you are preparing for any master's entrance exam or PhD entrance exam. That for that reason also you can talk to me if you have any doubt. And we will for this course I will be focusing on immunology, host pathogen interaction, and will clear your doubts. And this classes are tutorial classes. This so will be more or less interaction sessions, and we both will learn from both sides. Okay, I will start today's class. I will share my entire screen. Okay, just one error. Okay, some error is occurring. I'm just logging out and then logging in again. Just wait in the lobby, Malik. You don't sign out. I'll be back. Okay, so there was some technical glitch. I'm extremely sorry for that. We will start today's class. I have started sharing my screen. If you can't, yeah. Okay, I hope my screens are visible to you. My slides are visible to you. So we'll be starting with today's class. Mm. Okay, so as I was telling, so this is a tutorial classes for the course host pathogen interaction, and this is the week 1 tutorial sessions, and which is offered by NPTEL or SWAM portal. Course instructor for this course is Professor Himanshu Kumar from Department of Biological Science, Iser Bhopal, and I'll be one of the teaching assistants for this course. Ananya Chatterjee. I'm a graduate student in IIC Bangalore. So about the course you have already gone through the in the NPTEL portal. Still I will just go through because this is the first class. So this course basically introduces the fundamental aspects of host. Host means for our respect, like with respect to us, the host means us, like human beings. And also because we do experimental studies, so we can also do experimental analysis in mouse and other animal systems. So in that case we can also see mouse as a host. So we basically want to discuss the fundamental aspects of host defense system. What is defense system? You all are aware that defense system means whenever we are being infected with some foreign particles. It can be bacteria, it can be viruses, it can be other parasites. So what happens, we do have an defense system. Okay, so our defense system tries to nullify their effect. So what are the defense system? Main and the essential and the only defense system is our immune systems. Okay, so immune system, those consist of different things. Those consist of different immune cells. We will, as soon as we'll proceed forward for the courses, we'll discuss what are the immune cells and how, what is innate immunity, what is adaptive immunity. So our defense system basically helps us to fight against those parasites. So we will be discussing this stuffs in great detail in those courses. This is a 12 week course. So, and also we will discuss how our immune system develops immunity, okay, against foreign living and non-living entities as well. So basically this course, as I was discussing with you that in which courses you are, so this course is basically good for bachelor's, masters and as well as research level, because whenever you have started your PhD, you need to brush up your basic knowledge. So so this is another brush up and another learning for me as well. We will discuss and we will learn together. So from different disciplines of biological science, biotechnology, chemical science, medical science, to understand the basics of here host defense system. Okay, so we have courses in our bachelor's, we had immunology course, in masters also we had immunology course. Okay, so like that we will learn. So it will be important whoever are doing bachelor's and whoever are doing masters and planning to go PhD. In PhD also, if you are in lab which works mostly on like immune cells and immunology, you will again need to brush up this basic concepts. Okay, so this is a 12 weeks course, and credit points 3. This is sorry, anyone wants to tell anything? Okay. So this course has started from 22nd January and today is 30th January. We are starting tutorial classes and the course end date is 12th April. And enrollment for the course and registration has already be started. If you want to be appear for the examination, which is also a certificate course, this is a certificate course, so you will get certificate if you register for the course for examination. You have to remember the exam registration date ends on 16th February. So please, whoever has not registered but willing to register, do register as early as possible, and exam date will be 27 April. Okay, so my plan is that today week 1 we have started on 30th January. So each week we will be discussing each week's content. Like in week 1 I will be discussing week 1 content which is already been uploaded. Okay, please go through those contents whenever it's uploaded, and I will again go through and will discuss if you have any doubt. Okay, then like that, this is a calendar for us for next 12 weeks. So course layout. So week 1 course layout is the very basic fundamental of immunology, where it started, from where we got to know that yes, there is a defense system in our body as well, and we need to fight with the surrounding environment. So week 1 basically history of development of immunology field. Okay, so in biology there are several fields, so among them immunology is one of the most crucial field, you know. And then we'll be discussing different branches of immunology and an introduction to immune system, overall introduction to immune organ. And week 2 and so on like that. Week 2 will be immune organ, week 3 will be cells of immune system. So in the immune system, it's a kind of a diversified cells, it accomplished of diversified cells, right? So we will discuss on that. Week 4, basics of cytokines and other immune mediators. Okay, week 5, introduction of innate immunity and how innate immunity influence whole immunity. Week 6 will be innate immunity. Week 7 will be innate immunity and humoral components. Week 8, antigen. And week 9 will be brief introduction to innate immune responses during virus infection. We will discuss that when we get infected with virus, what our immune cells does, and how virus, sometimes we know that we have already been through the COVID-19 pandemic, and we know that how this virus severely affect us, right? So we have a defense system, but still this virus are able to just bypass the defense system somehow. So we'll discuss that how viruses invade innate immunity to establish infection. Okay, Week 10 we will be discussing immunological responses during some specific viruses. Here we will be discussing on influenza viruses, and we will also discuss brief introduction of viral infection. Then week 11 will be discussion on immunology of bacterial infection. And the last week we will discuss immune responses to parasites and fungal infections. So basically we will discuss host immune system as well as uh, with a respective with a respective view of pathogen interaction, how pathogen also tries to invade. Okay, so this will be a very interesting and enriched course throughout. So some books and references which is also already mentioned in NPTEL website, you can follow. Kubi's in Immunology for Kubi's is a very important book and very nicely written book, you can follow. Another immunology book is Jensen's. These are I can recommend you these books. And also here some recommendation as there, the essential immunology from Peter, immunobiology Genway, yeah, Genway. So as I was telling, Genway, immunobiology, and Kubi, these two are very important book. Okay, please go through, please have a look to the book if you want to understand more. Then here professor also will talk about different review and research articles, which I will also be talking whatever he suggests. And then there are another infectious disease pathogenesis prevention and case studies. So these two books are mainly one of the most important books in immunology fields. And also yes, there is, as I was telling, that there is course certificate, and exam date is 27th April. Yeah, please, if you want to register, do register for the course. And yeah, I'll be admitting some people. Okay. Okay, so criteria to get a certificate. Criteria to get a certificate. Average assignment score should be 25% of average of best 8 assignments out of 12 assignments. Okay, so you will be given 12 assignments. Okay, and out of them you will be counted for best 8 assignments among 12 assignments will be counted. Okay, and on the total marks of assignments, 25% will be taken for grading. So average assignment scores 25% of average. Okay, and exam score will be 75%. Okay, 75% of the proctor certification course among out of 100. So the total exam is like 100. 25 marks will be coming from assignments and 75 marks will be coming from your final exams. So assignment score will be averaged. So you will be eligible for a certificate only if average assignment score greater than equals to 10 out of 25. Okay, and exam score should be 30 out of 75. If one of the two criteria is not met, you will not get the certificate even if the final score is greater than 40. Okay, you have to keep in mind. So certificate will have your name, photograph, and so you can find this information in NPTEL website also. So as you have already seen that week 0, there is a week 0 assignment, which is basically the preparation for the course. So week 0 assignment is I will discuss with you. So we'll start. The first question was, what is the primary function of the immune system? I have already marked those correct answer, so we can discuss. If you have any doubt, please write down in the comment section. I will go through and I will talk about that. So options are, what is the primary function? Digestion, respiration, protection against infection. And muscle movement and blood clotting is very simple. It's protection against infections. And also another important thing is that every classes I am recording, like tutorials classes also I am recording, and the slides I am using I will be uploading in the document folder. So those folders and recording links will be shared to you guys by NPTEL team, and as well as it will be available in the NPTEL website. So if you miss by chance any classes, any tutorial classes, you can go through those recordings as well. The second question is, which cells are considered the main soldiers of the immune system? Which you all know, which is WBC or white blood cells. These are the main soldiers of the immune system. What is the role of antibodies in the immune system? Energy production, cellular respiration, signal transmission, defense against pathogens, blood clotting. It will be defense against pathogens. Which of the following is a viral infectious disease? Tuberculosis, influenza, malaria, syphilis, Lyme disease. So we all know that influenza is a viral virus. So and influenza is a virus, and also this is a viral infectious disease. Okay, so we also call the disease like we have got influenza like that, but the virus name is influenza. So what is the primary mode of transmission for the HIV? Is HIV airborne? No. Waterborne? No. Vector-borne? No. Sexual contact? Yes. Foodborne? No. So this is basically blood. It gets transmits by blood. Okay. So which immune cell is responsible for engulfing and digesting pathogens? Okay. Which immune cells is responsible for engulfing? So we know that macrophages. Macrophages do engulf and digest pathogens. What is the function of vaccines in the immune system? Cure ongoing infections, protection from infections for which vaccine is given, suppress the immune response, increase blood clotting, improve digestion. So what would be the answer? The answer is protection from infection for which vaccine is given. Okay. So what is the function of vaccines in the immune system? So vaccine basically gives a protection for the disease you are taking the vaccine, so that it gives us a immune memory. Okay, and then when we'll get infected with the disease, our immune cells can remember, like memory immune cells can remember and then fight against the viruses or whatever the pathogen is. And then which of the following is a bacterial infectious disease? So chickenpox no, tetanus yes, dengue virus no, measles mumps no. So only tetanus is a bacterial infectious disease among this 1 2 3 4 5 options. Okay, what is the function of the spleen in the immune system? So what is the function of spleen? Option 1, blood filtration and protection, digestion of nutrients, oxygen exchange, hormone production, muscle coordination. What will be the answer? Blood filtration and protection. Okay. Which of the following is a fungal infectious disease? Candidiasis. Candidiasis is a fungal infectious disease. Okay. So this was the week 2 assignments which we have discussed, and someone wants to be in. Okay, now we will go through lecture contents which is already uploaded. Okay, by Professor Himanshu Kumar. So we won't be going to each slide, but the basic thing, the brief overview I'll tell you, I will discuss with you, and so that the next day you can, if you have any doubt, please let me know. Okay, and go through and read, understand every slides which has been uploaded, because you need to understand, first of all you need to clear your concept, which is very important. Please feel free to discuss in the discussion forum, as well as you can ask me. Second thing is that if you are appearing for the exams, all assignments and all questions will be covered only from your slides, and if your concepts are clear, you will be able to answer. And some things you need to remember also, because in week 1 assignments, if you see, you will see that you need to remember some names like Nobel Prize winner and some other names, because these are kind of history part we are starting with. So here you need to remember those things. So we'll start. So this birth of immunology. So here it is written that the discipline of immunology grew out of the observation that individuals who had recovered from certain infectious disease were thereafter protected. So what was happening, that people were seeing that some people are getting infected, and but if they survive the infection, then afterwards if this kind of infection comes also in the territory, they are the protected ones. Those infections are not getting like those infections are not affecting them anymore. So people started thinking about it, so what is going on. So the immunology, so immunology is the subject is immunology, but the source of word is immunity. Okay, immunity. So this is a Latin term and it comes from immunis. This is also important to know. Okay, especially for bachelors who are studying the immunology course for the first time. So immunity word has come from the Latin word immunis, which means exempt or protect. Okay, so historian Thucydides, in his contemporary description of the plague of Athens, he stated that, which is here it was with those who heard recovered from the disease that the sick and dying found most compassion. They knew what it was from experience and had now no fear for themselves. For the same man was never attacked twice, never at least fatally. Okay. So this was the question, that how, what is happening that people are getting affected and the second time they are not getting affected fatally. So from where it all began, we all know that it all began with smallpox disease. It was a it was a massive pandemic around the world. So we have in the classes, professor has already discussed the history. Please go through those lecture videos as well, because I will only be repeating the same things. So this is a smallpox disease, and now we know that there is a virus which causes the disease, smallpox virus. So the symptoms, we all know that it's high fever, body ache, viral fever symptoms, and so these are the physical symptoms which we see, that rashes, and then some sores become pustules, and after some time, after the rash disappears, and this is a pathophysiological conditions. Okay, so and then people started identifying that from where it's getting affected, because that time there was not much knowledge about viruses and then immunity. So people just trying that, so what happens if we collect those pustules and infect other healthy individuals. Okay, so it started from all over that phase. So variolation means, so now variolation means that fluid from live smallpox. So one person is infected with smallpox, so from this pustule, people started collecting fluid and was injected subcutaneously into healthy individuals. So this term is variolation. Okay, so variolation then gets lead to vaccination in around 1798. So exposure to cowpox protected the individual against the more dangerous smallpox. In 1798, the first smallpox vaccination was notably demonstrated by Edward Jenner. You need to remember the name, that the first smallpox vaccination was notably demonstrated by Edward Jenner in 1798. So first method to be called vaccination. So around 1798 it was the first method after trying, and you can tell vaccination because you were injecting the virus, and then people didn't understand, but the thing is that whenever you were injecting small amount of viruses, our immune cells was able to fight against it and make some memory immune cells. Okay, so that the next time they are not getting affected. And after some time, global eradication of smallpox happened around 1979. First modern controlled experiment in immunology. So Louis Pasteur first vaccinated one group of sheep with heat attenuated anthrax bacillus anthracis. Okay, he worked on bacillus anthracis, and he vaccinated one group of sheep. He then challenged the vaccinated sheep and some unvaccinated sheep with a virulent culture of bacillus. All the vaccinated sheep lived and all the unvaccinated animals died. So Louis Pasteur, you all know, also called as father of immunology. In 1885, Pasteur administered his first vaccine to a human, okay, a young boy who had been bitten repeatedly by a rabid dog. So now we will look over Koch's postulates, which is very important in microbiology, immunology. So Koch's postulates was gave around 1875. So what are the points, important points of Koch's postulates? You will also see in your week 1 assignments. So remember, first option is the microorganism must be found in diseased but not healthy individuals. The microorganism must be cultured from the diseased individual. So you will be able to culture microorganisms if you collect those from diseased individual. Okay. Inoculation of a healthy individual with a cultured microorganism must recapitulate the disease. The microorganism must be re-isolated from the inoculated diseased individual and matched to the original microorganism. Okay. So these are 4 important postulates you need to understand and remember. Koch phenomenon and tuberculin skin test. It was happened in 1891. So Koch discovered that organism causing white death or tuberculosis. He also showed that tuberculin, a product of the culture of tuberculin, would elicit a severe local inflammatory reaction in tuberculosis patients upon intradermal application. The tuberculin test has proved to be invaluable in the diagnosis of past or present exposure to tuberculosis bacilli and set the stage for the development of numerous other diagnostic skin tests. So the excessive dermal reaction to tuberculosis bacilli in the skin of sensitized animal. Yes. This is Koch's postulate. So please go through those slides again if you want to. I will be uploading all slides, and these slides are basically from Professor Himanshu Kumar sir's class. So those are already uploaded in NPTEL website. So and then so today's class is basically history class. So then there the birth of immunology started, right? So okay, so this is another history of immunology part 2 will be starting. So professor has explained well that there are different cellularists and humoralists. So the chief defense of the body against infection resided in the phagocytic and digestive powers of the macrophage and the microphage. Humoralized, only the soluble substance of the blood and other body fluids could immobilize and destroy invading pathogens. So okay, so there is some mentors, opponents and collaborators. So there are a panel of scientists who gave some important discovery whichever they have done. So there are two kind of thing, innate immunity and adaptive immunity. So innate immunity means we have some immune cells, so those are cell or cellular. So there are two panels of scientists who are telling that some cells are important or chief defense system for the for our different immune system, and there are also some scientists have discovered that yes, there are another thing which is acquired immunity, where mainly humoral or soluble substance play a role. Okay, so this is what is stated in this slide. So they have some eminent scientists gave their reports. You can see that they have stated that phagocytosis as general mechanism of host defense, and then cells central to immunity, Radolf Virtue said, and then Julius said that phagocytes and inflammation. Emil Fischer talked about key lock principle for differential cell staining, sidechain theory, chemotherapy. Jules stated complement as mediator of antibodies. Emil has stated for principles of antigen antibody interactions. So these are different discoveries from different eminent scientists. So, phagocytic theory of immunity. It came around 1884. So, he placed thorns in starfish larvae. The amoeboid cellular entities that seem to engulf the thorns. So, Mechenkov termed such cells phagocytes, giving rise to the field of cellular immunology. So Mechenkov hypothesized that cells rather than serum components were the major effector of immunity. So there are different, you know, hypotheses and statements you can go through. Ehrlich, physician with eyes of a chemist. So he has stated that union of toxin molecules with side chains of the cell protoplasm, the antitoxin, utilizing their haptophore groups, cells under the threat of foreign microorganism grow extra side chains to capture the toxin elements. These additional side chains designed to break off into the circulating blood flow were identified as an antibodies. The first model to propose for an antibody molecule in which the antibody was branched and consisted of multiple sites for binding to foreign material known as antigen. Isolation of diphtheria toxin was done by Emily and Alexandra. So, using the key studies of German pathologists Claves and Rolfer, who had isolated the diphtheria bacillus and demonstrated that it causes illness, Ursin and Ross have found something very interesting. The bacillus itself does not enter a victim's bloodstream. Instead, the bacillus produces a poison or toxin, which is the disease. So, bactericidal power of blood. George Nuttall, defibrinated blood possessed a considerable bactericidal power against anthrax bacilli, and this power was destroyed by heating to 55 degrees Celsius. And what Dr. Buchner said, Buchner was one of the first to note that a substance in blood serum, alexin, could destroy bacteria. Immune bacteriolysis or fever phenomenon. The fibroids inoculated into the peritoneal cavity of normal animals rapidly multiplied, but the same vibrios in an animal that had recovered from the infection and underwent bacteriolysis, that is, they rapidly swelled up, dissolved, and disappeared in 20 minutes. This took place without any cellular activity whatever, and in fact, the serum of an immune animal produced the same lysis in the test tube. This is the Pfeiffer phenomenon, which is still in use of differentiating closely allied species. Bactericidal power of blood. Serum as a protection matrix. It was dated around 1890. So I'll go over those slides quickly. Bacterial agglutination. Bacteria of similar size clump together in sera in ways specific to or determined by each serum. The first crucial practical consequence of Grover's work on bacterial agglutination occurred in around June 1896 when the French physician Georges developed a diagnostic agglutination test for typhoid, therefore known as Gruber-Weidel test or Gruber-Weidel reaction. Okay, the typhoid test reaction is called Gruber-Weidel test or reaction. uh, so this is the scientist who won Nobel Prize in the field of immunology. So these are their research domain, and which on which domain they got Nobel Prize. So there are several branches of immunology. Advances in the immunology has led to the emergence of the lot of studies of subspecializations apart from classical immunology. So there are different branches of immunology apart from classical immunologies. Those are clinical immunology, cellular or molecular immunology, cancer immunology, transplantation immunology, development and comparative immunology, immunoinformatics or computational immunology, mucosal immunogenetics, neuroimmunology, structural immunology, reproductive immunology, nutritional systems immunology or vaccinology, viral immunology. Okay, these are different interesting topics which comes under classical immunology. And also there are another immunology like subclass which is clinical immunology, deals with the diagnosis and treatment of diseases related to dysregulation of immune system. So immunodeficiency disease, what happens when our immune response gets suppressed, that is immunodeficiency. And excessive immune response happens when our own immune cells detect our own cells and then fight against our own cells, which is autoimmune disease. Okay, where excessive immune response happens, but immune system needs to be balanced, needs to be in homeostasis. So, cellular and molecular immunology deals with interaction of the cells and molecules of immune systems that are involved in the recognition and elimination of pathogen. Okay. Cancer immunology deals with the understanding of role of immune system in the cancer progression and development, as well as treatments such as immunotherapy. So immunotherapy is very crucial, one of the very crucial treatments against cancer, where we boost our immune cells. So we need to understand the role of immune system in the cancer progression and development as well. Transplantation immunology deals with the transfer of grafts or transplants from one individual or animal to another individual. Autologous, when you were getting transplanted one organ to your own system, that will be autologous. Syngenic, allogenic, xenogenic. Okay, syngenic means it's it's like a little bit similar. Allogenic means different, and xenogenic means different species. Okay, syngenic means we will talk about it like in relative kind, in between relatives, where there is chance of graft rejections but it might be less. Allogenic is different, and then xenogenic means from different species. Development and comparative immunology deals with the analysis of shared or diverging facets of immunology among different species. The comparisons are carried according to the principle that evolutionary conserved themes outline biologic functions in universally relevant for survival. Immunoinformatics or computational immunology deals with the conversion of available immunological data into meaningful mechanistic interpretation using computational models or algorithms. Immunological data, high throughput sequencing, microarray, genomics, proteomics, hypothesis generation, validation of hypothesis, mechanistic interpretation. Mucosal immunology deals with the study of immune system responses that occur at mucosal membranes of the intestine, the urogenital tract, and the respiratory system. Immunogenetics deals with the understanding of relationship between genetics and immunology. The genetic variance of certain regions in the individual can lead to dysregulation of immune system and affects the susceptibility to certain disease. Okay, so it depends also on gene and what region you are, and so that you will be like susceptible to some certain diseases with respect to that. Okay. Neuroimmunology deals with the study of the interaction of immune system and the nervous system during development, homeostasis, and response to injuries. And then structural immunology deals with the study of structure of immune molecules, antigens. If you need to know the what is the structure, so this immune like those molecules, antibodies and all those all are proteins, so you can solve those structures. Okay, so if you want to know the structure, the how antibodies or antigens look like, those fields, like you can, there are several structural biology field, like NMR, crystallography, then cryo-electron microscopy. Okay, you can do those structural biology, take those structural biology approaches to solve your desired antigen or antibody structure. Even interaction also you can see. So deals with the study of structure of immune cells, antigens, antibodies, pathogen receptors that can be targeted therapeutically for the infection treatment. Structure of various human virus proteins crucial for the pathogenesis leads us to the development of therapeutic and vaccine candidates. Reproductive immunology deals with the understanding of immunological relationships between developing fetus and mother immunity. B cells in physiological pregnancy, B cells in preeclamptic pregnancy. So we will talk about it in our future classes. Nutritional immunology deals with the understanding of influence of nutrition on the immune system and its protective functions in the interface of host pathogen interaction. Systems immunology or vaccinology deals with the system level approach to elucidate signaling pathways that are important in the immune system and create a model of how different cells of the innate immune system function. Another approach of system biology focuses on the cells and the molecules and how they respond to vaccines, infections, and so on. So system biology approach are also there to understand like vaccines with known correlates of protection, and then vaccines with unknown correlates of protection. Okay, viral immunology deals with the study of virus and immune system and the complex host pathogen interaction. Both the DNA viruses and RNA viruses induce distinct immune responses. So these are the introduction which we will go through in our future classes. Now, an introduction to the immune system. So there are different sizes of cells, from bacteria to very small viruses, bacteriophages, unicellular parasites, large parasites. This is a scale bar where you can see bacteria around 0.5 to 5 micrometer, bacteriophages around 20 to 200 nanometer. Viruses are very small, can be 100 to 800 nanometer uh, in diameter. Unicellular parasites a bit bigger, 10 to 30 micrometer, and large parasites are around 0.2 to 8 meter. So these are different diversified parasites, which some of the parasites you won't be able to see through your eyes. You will need electron microscope, which is viruses. Some of the like things, bacteria, you can easily like visualize under light microscope as well, and some large parasite you can visualize through your eyes. Also, along with size diversity, there is also biochemical diversity, like how does those thing are made of. Chemical composition, that is protein, sugar, lipids, or complexes of various biomolecules with huge size and weight variation. So there are variation. Specificity, broad range of specificity, lipopolysaccharides from various gram-negative bacteria, flagellin from various gram-positive bacteria, beta-glucan from various fungi, strict or narrow range of specificity, aminobenzene, alpha-aminobenzoic acid, aminobenzoic acid, meta-aminobenzoic acid, and para-aminobenzoic acid. Properties and hallmarks of immunity. So these are example: systemic infections, these are some virus family, and persistent of antibody, and these are mucosal infections. Okay, so one example you can see that chikungunya, which is alpha virus, and persistence of antibodies around 30 year, and covid, which is coronavirus from coronaviridae family, persistence of antibody around 1 year. Okay. Discrimination among self, non-self and modified self. So host own cell is self, multilayer of uninhibited cancer cells which is like modified, and then non-selves are virus, bacteria and parasites. Tight regulation molecular level, so transcription, post translation, transcriptional non-coding RNA regulators, translational level, positive or negative feedback system, pro or anti secretions, cellular level, suppressor cells, M1 M2 macrophage T regulatory cells. It's a tight regulation, so we will discuss in detail. So we have innate immune system, adaptive immune system. We will discuss about development of immune response, elimination of pathogens. There are cell components or non-immune or immune cells, which is related with pattern recognition receptors. Pattern recognition receptor will detect pathogen associated molecular patterns. We will discuss about this. So basically pattern recognition receptors resides on immune cells, so those receptors can recognize some specific molecules which are present on pathogens. Okay, and those specific molecules has also a pattern, so it's called pathogen associated molecular patterns. So those initiate inflammation, induce type 1 and type 3 interference and activate innate immune responses. Initiate the pathogen specific adaptive immunity via B and T lymphocytes. So we have several important organs for immune systems. One of those is lymphoid organs, sites for the generation, maturation and initiation of adaptive immune responses. Organized tissue in which lymphocytes interact with lymphoid, non-lymphoid cells and trapped antigen. Primary lymphoid organs, immunocompetent cells are generated and mature. Secondary lymphoid organs, organs where interactions of antigen naive immune cells take place, present in path of lymphatic vessels, reach in lymphocytes and macrophages. Prenatal lymphoid organs or system, embryo yolk sac, 1 week of development, fetal liver 3rd month, spleen 7 month. After birth, your lymphoid system will be bone marrow. Thymus, anterior superior mediastinum, bilobed organ on top of heart. Okay, pinkish gray colored soft tissue which is weighed around 30 gram in infants and 3 gram in adults. 95 to 99% of T cells die in thymus. Self reactivity or no reactivity to antigen by process of clonal selection or clonal detection. Cross section of thymus. So this is a cross section of thymus. You can see the dividing thymocyte. Thymocyte means the cells of thymus is called thymocytes. Trabeculae, capsule, dead cells, narcels. So these are Hassel's corpuscles. So the thymus are rich in dendritic cells. So interdigitating dendritic cells are there. Macrophages, all immune cells are there. So consist of cortex, immature T cells and densely packed, and medulla which is not densely packed. So cortex is like the outer region and medulla is inside. Dendritic cells, macrophages and epithelial cells important for growth and development of developing thymocytes. Okay, and which is a thymectomy, which is the like if there is a dramatic fall of T cells in your body is called thymectomy. Okay, in human Dijor's syndrome is also called. Site of B cell generation along with the other cell, so it's the bone marrow. Okay, with advancement of age it is mainly filled with fat, around 50%. So we'll be discussing different important cells like hematopoietic stem cells. From there you will have lymphoid cells, pro B cells, pre B cells, then immature B cells and then mature B cells. So basically the development of B cells in bone marrow. Peripheral lymphoid organ, so then B cells comes in the peripheral lymphoid organ where differentiations happen, and it will be differentiated in plasma cells, which will followed by which can be able to secrete your antibodies, and we will have memory B cells of various types and plasma cells secreting various isotypes. So we will discuss in later classes. bone marrow, it is not a site of B cell maturation in all species. Example, in birds, bursa of fabricius is a site for B cell maturation. In cattle and sheep, fetal spleen, Peyer's patch, and in rabbit, appendix. Again, this is spleen. Spleen is state here, just beside pancreas. So it's a cross-sectional view of spleen. So it has, these are capsules where all those internal structures are there. So it spleen filters blood, traps blood-borne antigens. Splenectomy in yonks leads to systemic infections. These are primary follicle. These are marginal zone. Periarterial lymphoid sheath, or it's also called pulse. These three together also called white pulp. Germinal center, artery, and this is vein. So this cross section also, if you are in bachelor's, you need to you can just draw, and then you can practice that where those trabecula are, and then where vascular sinusoids are, to know. Lymph nodes. So there are a lot of lymph nodes in our body, in this region, in here in retinoperitoneal regions. Okay, so those are encapsulated bean-shaped structures, contains networks of stromal cells packed with lymphocytes, macrophages, and dendritic cells, connected to both blood vessels and lymphatic vessels. Cross section of lymph node. So here this is afferent lymphatic vessels, which afferent means uh, those are products are going inside from those vessels. Yes, Parth. You can, if you have any doubt, please write down in the comment section. This is afferent lymphatic vessels, and these are primary lymphoid follicles. Again, this is capsule where those follicles are. Germinal centers, B lymphocytes, lymphatic artery, lymphatic vein, efferent lymphatic vessels, postcapillary venule, germinal centers. So multiple afferent lymphatics. Cortex consist of B cells, follicular dendritic cells, macrophages, germinal centers, paracortex T helper cells, macrophages, and dendritic cells. Medulla consists of plasma cells. Post-capillary venule allow lymphocyte migration from circulation into lymph node. Only one efferent lymphatic. So you need to understand that there are several afferent lymphatic vessels, but there is only one efferent lymphatic vessel. So rich in antibodies and lymphocytes. Lymphatic system. So these are lymph node, these are lymphatic vessels. So sub part of blood vascular or cardiovascular and immune system. Plasma from blood seeps into tissues. Okay, so these are tissue space, lymphatic capillary, lymphatic vessels, lymphoid follicles. Lymphatic system. Thoracic duct is largest lymphatic vessel, empties into left subclavian vein. Okay, thoracic duct is the largest lymphatic vessels which empties into left subclavian vein. Movement of lymph in lymphatic vessels depend on muscle contraction. One way valves ensure one direction. Lymph nodes act as filters for antigens. So just a second. Okay. So we will end with lymphatic system today, and we will discuss another other immune cells in detailed manner from next class. If you have any doubt, you can let me know. We will discuss today. So if you have any question, okay. Yes, Parth. Okay. Are lymph nodes present in brain? So you can see, Parth. As far as I know, the brain is devoid of lymphatic systems. So far we know mostly lymphatic systems are present, in the thoracic region and as well as the peritoneal region, retinoperitoneal regions. And also you can see in like the lower part of the body, you can see those lymphs. But in the central nervous system, we are not aware of. Yeah, we are not aware of.
Okay, but the question is artery and veins are attached by lymph nodes. As my knowledge,
Attached what?
arteries and veins are attached by lymph nodes in circulatory
Yes,
systems.
yes, yes, yes. So there is a junction between arterial veins with lymph
Uh,
nodes where fluid exchange happens. So there is blood-brain barrier. But about the lymph nodes, I need to look into details, because as far as I know, central nervous system is devoid of lymph node. I will look into it. You can
okay,
also,
okay.
yeah, yeah. If if you have any other question, we can discuss, and we'll discuss about those doubts. And also, yeah, you can share the PPT. Yeah, I will share the PPT. The PPT will be available from your NPTEL website. You can revisit the download, like this tutorial sessions as well. The video also I'm downloading, and it will be uploaded in a YouTube channel, which is NPTEL lectures by Ananya. You can see in that YouTube channel, and link will be shared in the NPTEL website. So today's class was basically an introductory class where we have learned about different immune systems, immune cells, and where those are present. In next class, we will be again talking about other several other immune cells and immune systems, and lymphatic system. Till lymphatic system we have learned. So, any other question? Yeah, I will send the PPT. You can find it in NPTEL website. I will be uploading my
What will you send it?
in so I will so in NPTEL website you can see that download lectures. Okay, in those download lectures you'll be able to download PPTs, and also the tutorial session, like sessions whatever is going on, those tutorial lectures slide, like the presentation slides as well as the lecture videos. This class videos will be shared. You can get access through that. Okay.
Ma'am, sorry. Where they find PPT?
Can you guys please write down? I'm not able to hear your voice actually. It's network difficulty.
Sorry, ma'am. Where they find PPT?
PPT NPTEL SWAM portal. You have to go in NPTEL SWAM portal. You will so in your course website, na, there is a web portal. In that you will search host pathos interaction, where you are getting all your like week 1 week 2 contents, and there you will have one download lectures link. Okay, just go through the links and then you will have all
Okay.
presentation slide downloaded. Yeah.
Thank you.
Will mail be sent for each tutorial class link? Each tutorial class will be the same link. So today's link is the same link for next 12 weeks class. But yeah, mail will also be sent. But this is the universal link for every classes. If you don't have any doubt, we can stop today's class and we will again meet next Tuesday, 6 to 8 p.m. So, if you have any doubt, please feel free to ask in this class, and also write down in the comment section. We will discuss. This lecture will go per day and same time. This lecture will go each week, every Tuesday 6 to 8 p.m. Yeah, same time. Okay, thank you all. Thank you all for joining. We will see again. We'll meet again next week, Tuesday, at 6 p.m. Bye.